Welcome to our third GCAN webinar. It's our honor and pleasure to have Dr. Jose Martin Hernandez-Ayon joining us. I'll just, well, I think we kind of broke order a little bit. So let me do the introduction, and then please be mindful that this webinar will be recorded and it'll be later published on the GCAN's website. So Dr. Hernandez-Ayon is oceanographer with the doctoral studies in coastal oceanography and a postdoc fellow at Scripps Institution of Oceanography in UCSD. So he's a specialist in the carbon dioxide system in seawater and marine biogeochemistry. His research has focused on studying the role of coastal zones in the carbon cycle, including the effect of ocean acidification on marine ecosystems and the relationship of ocean acidification with other stress factors, such as hypoxia, climate change, and CO2 fluxes in coastal regions. And he has served as the chair of the Mexican Carbon Program since 2018 and is a science member of the Ocean Acidification Observing Network, or short name is GCAN, Executive Council, and a member of the North America Carbon Program Science Leadership Group. He's also a GCAN Latin American Ocean Acidification Studies Network LOCA representative. So without further ado, Martin, the floor is yours. Thank you.
Thank you so much, Jinping, for the introduction, and also Shoba for the invitation, and also for the organizers, GCAN community. And now I can show that the slogan is also changing, related with the Gulf of America. But it was interesting because I will be part of the transition to Gulf of Mexico to Gulf of America. But as we know, these territorial waters, Cuba, Mexico, and United States, we share this beautiful environment. And my talk relatively will be focused more on the territorial Mexican waters. But it will be nice, interesting to just that we know that we share this nice environment that's really with the Gulf. Let me tell you a little story about this, because I just pick up this interesting and historical plot from 2005. Ensenada is located in the Pacific side, and historically I work a lot in the Pacific sides. In 2005, we have a meeting in Seattle, and Dr. Richard Feely, Chris Vine, and also Dr. Takahashi I met in this nice meeting. The idea was to understand the influence of the upwelling effect in the ocean acidification in Canada, Mexico, and United States. I was happy with the meeting, but at the end of the meeting, Dr. Takahashi have a present for all the participants, and this guy give us a CD with all the data available for North America. I was, of course, happy to see that the Pacific side have data for the Mexican waters. But what the first time that I saw the Gulf of Mexico with the empty data, as you can see here below the latitude 25, we have no any data, just maybe in the entrance from the Yucatan. We are talking just a surface PCO2. The people from the United States know more about all the historical data available for that. But at least this map for surface PCO2 highlight our situation in the Mexican part. It was interesting to see that, because I was thinking, oh, we have a lot of homework to do in this area. 5 years later, when the BP, the British Petroleum catastrophe occurred, was interesting part because the Mexican government called the institutions focus on the oceanography, to work together and to see if any impacts happen in the Mexican territorial waters. But in reality, we don't find nothing. Was the truth? We were out there 6 months later. And the nice part of this moment was that the government, Mexican government, in reality, I guess, was motivated based on the presentation from my colleagues about the needs to develop a baseline, first baseline in several aspects: the biochemistry, the physical oceanography, the models, to know and to improve our knowledge about our territorial waters. Then we have a very amazing help to work on this area. Our institution, located in the Pacific, will move to be working for 5 years in the Gulf of Mexico, supported by our Mexican NSF, we call CONACYT, and also by the Energy Secretary from the government. We developed a consortium, a network, many people, about 200 researchers. We were working over there in different aspects. And then we were part, me, in this consortium. Something we did, and my focus on my talks will be focused on some aspects of the more coastal areas, like the Peninsula of Yucatan, when we did some measurements over there. And most of my talk will be focused in the 7 cruises, what we learn about that in different years in the more oceanic area in our territorial waters, but also some measurement we did in this part, north and south, close to the land in the Mexican waters. We did measurements of DIC and alkalinity in my lab, and my focus, my talk will be focused, about the CO2 system aspect. I would like to highlight these works because, as I mentioned in my introduction, to learn about the Gulf, we need to be working together, the 3 countries. And now jumping from 2005 to just a few years ago, I guess 3, this paper was published, and you can see the difference. Of course, US have a lot of work. You guys have been working a lot with very good results in your territorial, but I just highlight the area where we will be talking today. I like this paper because we work by the lead of Emily Jinping, Emily Hall, Kim Yates, Jennifer, Letty Barbero, many others. We were working very nice at this work, and I would like to use today in this talk as a roadmap, specifically for the Mexican waters in the Gulf, because the drivers that we highlight in this paper, of course, was related with the freshwater effects, the nutrients, the rivers, the ocean warming effect, the circulation, recent times, and other extreme weather's events. But in the Mexican part, in reality, what we have is a few from all the list, and I have clear a lot of work that we need to do. And something that I would like to share with you is some information related with, first, in my few slides, I'm going to talk a little bit about the riverine influx in the coastal, the submarine groundwaters that we have from Yucatan. My talk will be both related with the karstic areas, and finally, most of my talk will be related with the factors what we are learning about the circulation by the loop current, because we are learning about that from the 7 cruises, the effect of the loop currents, and the effect of the ocean circulation here in our water. This paper was published just years ago by my postdoc, Linda Barranco, and we did measurements in the Yucatan, which is a karstic area. But we saw in these baseline studies two phases. One, with the signal from the groundwaters discharge nearby the land with high alkalinity, high DIC, but because the city is increasing and they have a problem with the manage of the wastewaters, we observed values very close to the coastal, to lowest than 7.4, PCO2 very high, higher than 1,000, and subsaturation nearby the spots of these submarine groundwaters. And this was amazing. This is why I call local OA, because it's an effect of the population. But the other phase was when the loop currents is stronger and go close to the peninsula. We have a totally different phase, because apparently clean this new Caribbean water over the shelf, all these human remnants nearby the coastal. I know talking that this is good, but this is what happened, the advection control. In reality, we have the homework with the governments to be working in this problematic situation nearby the coastal and this karstic area, which is a challenge, because everything is karstic, and then all the manage of the infrastructure is a big deal in this area. Okay, this is in the shelf, Yucatan. We want to move a little south to Campeche, which is we have now a different phase, a different situation, also karstic. Also, there are groundwater. We can see in these plots the signal of the Candelaria River and Palisada River. We have these two transects. This was part of the a chapter from Jose Andres, which was a PhD thesis. He published this paper just last year under the advisor of Gilberto Cardoso, that is also part of the community, John Albert. And we have the opportunity to measure these samples in my lab. Okay, this guy divides river. You can see in these plots the part of the river. He called estuarine, the mixing between the ocean and this water. And he called the coastal water more offside. And then if we see these first plots, is the salinity effect during the dry and green rain season, and then we have the rainy season. And then this, I was mentioned that in this case we have a different situation, because we can see the signal over groundwater with high different salinities. From the river, we can see the effect of this groundwater with very high alkalinities, more than 400, high DIC. But the other phase is when during the rainy season, because during the rainy season we have a different phase. DIC and alkalinity decrease, not to the normal oceanic value that we are familiar with, still high. But the situation in this case is because the raining is moving organic matter from the land with where we have a respiration effect, and then we have a decrease on pH and a decrease in the aragonite saturation of omega aragonita. We have different values that can be lower in some situation than 1. This means that we have subsaturation in some conditions. This is why, again, we are talking about local OA for these karstic waters. And then in reality, we have a few measurements over there, and this is why we consider just a baseline for these coastal areas. We have a lot of homework when I was talking about the roadmap that I mentioned before. because there are other rivers that are just in the north part of the Gulf of Mexico when we don't have any data about that. For the moment, I just highlight these two topics, the Linda Barranco from 2022 and this paper from the last summer. We move a little bit to the circulation loop current effect, because we have a little more information about that. We highlight some of this topic in the paper, but I'm going to go deep about that, and I'm going to tell you something that we are just working the last couple of years. And then we move to the loop current effect. Something that I mentioned is this is our territorial Mexican waters. And then as you can see, if we imagine the loop current moving inside to the Gulf, and we again compared with the map with our territorial international waters from Mexico, the successful to take samples from the loop current is just a few, okay, directly. I mean directly because the territorial, and then we have this limitation to really target how the water is go into the Gulf, thinking like a coastal lagoon and asking the end members how the water and the biochemistry go inside to the Gulf. We learned that we need to understand as a chemist the physics, and this is another PhD student, Antonio Delgado, worked for our group, and he was working more on the physics. He was working just with the average of 20 years of data for the chlorophyll satellite products, and also for the absolute dynamic topography. The idea was to use these satellite products just as a sentinels of the Caribbean water, to have an idea, a statistical idea, that when we have the more incursion of the data of the low currents inside to the Gulf, and when it's minimum. And then what we found is with these two products was the maximum enter of the low current statistically is in summer, fall, the minimum late fall and winter. Both product was very interesting because in reality we were not focused on high chlorophyll or low chlorophyll. In reality, we developed a reference. For example, in this case, for the chlorophyll, we consider concentration of 0.08, more oligotrophic signals is looking like a blue color in reality, and also we took a reference of the high of the ADP to match this part and to see to do this estimation, because it's an estimation, it's a statistical estimation, and we found again the maximum incursion during this month, but also we did a gross calculation about the area of cover of these Caribbean waters. The highest is about 20% of the Gulf is covering for this Caribbean water inside. The idea was to have an idea about the average of that, how cover these Caribbean waters inside to the Gulf. An old paper from the 90s also teaches this net flow. The idea from this paper, nice paper, was okay. This is what the physical oceanographer said about where the waters come from. Because again, as a coastal oceanographer, I was thinking where did the waters come from and which is the biochemistry. It's coming mostly from the North Atlantic or Central Atlantic, okay, or it's coming from the Caribbean waters. And the calculation from these guys, considering bed groups that are millions of cubic meters per second as a flow, they say this is about 40%, 50%. The red ones represent the Caribbean, the green one represent the North Atlantic, And for this calculation for that year, they suggest a bigger volume is coming from the North Atlantic. But what's interesting because as you will see, our data tell us something, a different story about that. What we did was, because the difficulties to sampling in a different area outside to the Gulf, we will be analyzing the data that we got with taking a comparison with other cruises. For example, we take this from GOSE, the World Ocean Circulation Experiments data. We have the depth, we have the dissolving organic carbon, and in colors, we have the latitude. And then in this area, I taken this data from the North Atlantic, and you can see how is the vertical structure of the DIC. If we move to the south, I mean to this area, to the Caribbean waters, as I indicate here, on the same cruises, we can see the colors blue to pink are located over there, over here. It's high concentration, but also in our data indicated in black, we can see that inside to the Gulf, for the station where we measure, are more quite similar to the Caribbean waters, which is interesting this part because we are not looking 50-50, 50-40, and I guess some new calculation need to be done, probably because I'm chemist, physical oceanographer, and probably there are a new calculation about that. That's something that we need to refresh. As you can see here, one of the messages is our data inside to the Mexican area and the Gulf is more similar to the sources of the Caribbean waters. And I want to highlight just that below 1,000 meters overlapping, but we have a different story in the first 700 meters because we have the variability that we will be talking later about that. Another variables, my friends and colleagues, Dr. Victor Camacho, he was in charge of the nutrient analysis, nitrate, phosphate, and silicate. You can see the difference between inside the Gulf. He took that as a reference, as a North Atlantic, as a signal is different. Here the differences, 5.3 and 12 micromolar differences between outside and inside. And he compared the Clyber as I did for our measurements, and we see a different story. Agreement with my comments about the DIC variables. And then we were happy with this for the moment to see that, to know that the water mostly is coming from the Caribbean, not only for DIC, but also for nutrients and the other variables too. Okay, I just highlight the question if new net flux and bedrooms need to be done. Inside to the Gulf, we happy with the sources, you know about the sources, something that Gabriela Cervantes Diaz published in this paper, 2022, was related with these first 700 meters, when we have Caribbean, the different water masses. What I mean is the Caribbean surface waters, the subtropical underwater, and the Gulf common water, and also the tropical Atlantic central waters. Yes, we are talking about the first 600 meters when we have the variability, because we have the mesoscale, the eddy current. Okay, what she found is looking every profiles was that profiles CTD data nearby the loop currents, look the red ones have this shape with the water masses, and the western sides of the Gulf in the west, we can see that the subtropical underwater disappeared. Okay, just looking the physical oceanographer, but we were concerned about that, the implication, why did this occur, and why what's the effect if we have water with more respiration processes mixed and the effect and the distribution inside of the Gulf. There are a good report from Gentil, colleagues from CCS, 2024, published just last year, talking about a nice study how this is dissolved and the effect in the Caribbean water. I will be talking more about that, about this paper at the end. But we measure also DIC, we published in this topic, and we notice how in the red station we have the DIC is have a low concentration in the waters, and it's increasing in the western side, probably for the more recent time and respiration, but we will see more about that. Again, now what I did was separate all this data, looked at, we are using also the GOMEC from Leti Barbera data, because we don't have too much about the loop current, as I mentioned before. And what I'm going to show you here is, this is the longitude, okay, to separate the data from the west from the east. Again, the red ones and the arrow here represent all the vertical structure from the, this is the apparent oxygen utilization. That means more oxygen is available in this area when the loop current is going inside to the Gulf, but more respiration effects is in the western side. And we can see the separation, the variability again in the first 700 meters. And if we compare the west versus the east, now we can see the dissolving organic carbon. Again, the same structure. We have low concentration when the loop current is inside, and increase that dissolving organic carbon is about 50 micromoles, about between 30 and 50, okay, inside to the Gulf. Maybe because respiration effects, we didn't did special respiration measurements inside. Okay, this is the AC, and this is the omega aragonite. We can see again the red one spots is they have a higher values, the lower values is in the west part, and below the 600 meters is when we can see values nearby to 1. But in the west side, in the first, we will say that 400 meters is when we have values lower, and then we have the increase in the upper waters. But you can see the difference between the vertical structure for DIC, oxygen, or the upper oxygen utilization. We can see the differences in the biochemistry between the waters go inside and the special distribution inside. Something that I want to comment here is, of course, because our sampling don't have the target to monitoring the eddy currents, loop eddy currents. Of course, we target one station, but this is not strictly like that, as we will see later. But we can see the variability inside in the Gulf, and how originally the water is coming from with more signal from the Caribbean waters. Okay. We have the opportunity to be working when we have these loop currents events with the Olympus and Nautilus, and also another phase winter when statistically the loop current decrease. We probably have for example, this LCE is the jumbo, more almost at the end of the life. Okay, And then we have these two phases, but something that I just highlight is that is not strictly strict to see that separation of the east versus the west, because this water have properties that is moving to the west, and we can detect it, for example, the soup and this kind of structures inside. We did some water mass fraction analysis with OMP, something where we can, this is a very good approximation to see the percent of the water for the Caribbean waters, the soup, for even water, the soup, the common water, and the TAC. What we expect, of course, is in this structure, we can see the transit is pumping water from the loop currents. We can detect the soup also in the west, is what I'm talking about. That these processes are pumping characteristics directly to the west, but the other phase is when we don't have new eddy current loops moving to the west, because you can see now the signal of the soup is almost done in all the column of the water. And then it's interesting because of Gentil et al. Explained there are dissolution, and this dissolution of the soup, they become the Gulf common water in this area. But this is from the physical point of view. The question is, what about the biochemistry about that? But this is a homework that we have. This is another exercise with the same structure that what I mentioned. I just want to highlight our calculation of the DIC, and this is the estimated, but using the OMP models. And really, the physics is quite similar. Just in this case, this kind of model tell us how the physical, how strong is the physics to move the chemistry. And then we did with these conditions, and we did for this other condition where we don't have the soup. And something that we learned was how the DIC is modulated by the upper waters. Is this low concentration of DIC coming from the loop currents, moving from these structures, is decreasing. Of course, is modulates these the nutriclines and also the carboclines in the Gulf of Mexico. This is another example that we did. We did a separation, in this case, from these three structures. You can see in black correspond to this vertical structure, and blue is all the station outside the mesoscale structures, and the red ones, the few stations that we target directly in the eddies. And you can see something that we mentioned is related with the more oxygen, is related with the station that we just catch up the loop currents or the structures, but is low oxygen is outside when we have the dissolution of the soup, and we have the formation of the Gulf common waters, but also the effects and the dissolving organic carbon. And then it's, in reality, as I mentioned, it's a preparation, it's a paper that is in preparation by Cervantes Diaz to tell us the role of this information that we have related with the modulation of the nutricline and the carbocline inside. Okay, and just to finish this talk, I just would like to highlight this new project that we are been working the last few years. We call Fitobloom. This Fitobloom project in reality is a multi-platform. This we have the opportunity in reality to catch up an eddy, and something that we did was to collect samples with a lot of details work with these guys and this Eddy, that people call Yasu, okay, the Woods Hole group, they call Yasu, and we did some biochemistry measurements in a transit that we did. This guy from the CCC, led by Enrique, Julio, and many other physical oceanographers. They use gliders to monitor this structure, but my colleagues, Victor Camacho, Orión Orzagaray, and Guillermo Samperio, we did measurements about the chemistry of this structure. The idea was to know, of course, to learn more about the processes inside to these interesting structures. These guys, the physical oceanographer, just published last year this nice paper, and this guy just highlight how these structures they call warm core rings reshape the water masses and distribute a large amount of heat and salts. And what they found was below the mixing layer, they found some increase of the eddy edges that is driving heat and salts. They call also all ocean chains to the surface. It's interesting because what you see here in this structure, they describe which water masses are inside, and they divide the central part and also the edges, but they highlight the effect of the edges moving this water to the upper part by observation of turbulence, and they call submesoscale processes that these are stirring of the spicy gradients that are transforming the subtropical underwater in the Gulf Common waters. This is something that is available, but this base help us to understand a little bit about our targets as a biochemist. That you can see here is the Yasu, and these are the stations that we collect. Something that these satellite products help us to see is the effect of the Mississippi and this area just by temperature, but also we notice a little pulse of low salinity in one of the stations, but also chlorophyll in this area. You can see the extension of this structure, but also the challenge to understand the area, the effect of the biochemistry. In my group, Orión Orzagaray is working this paper preparation because we set up an underway system for PCO2 and pH. Of course, we use the measurements and the 17 transits in the first 500 meters for total alkalinity, pH, and dissolved organic carbon. But in the surface, you can see the structure of the salinity from the thermosal. Okay, looking, this is from the east to the west. This structure, we can see the pH is low in this area and the edge of the east side, but also more PCO2, low pH. And of course, he did some calculation about the fluxes based on using a fixed alkalinity for all this structure and using the pH. And he estimated values about 4 millimole per cubic square meter per day as a flux for this area when we have the low pH. And then it's not homogeneous. Homogeneous is the central part, but the edges is when, as Gentil et al. Explained, we have this dissolution of the soup is pumping salt and this water to the edges. This is the TS from this structure, and we can notice how the soup is over there, but also we have some dissolutions transit that we found at the edges. What we can see here is a transit west by longitude, And then this red one and blue one represent the two edges of this structure. When you can see the salt pumping to the surface because it dissolved in the edges of this soup water with these biochemical properties to the upper part. And because this is interesting because we can see, we can see the temperature is cold more in the east. The DIC was with high concentrations about 2100, more close to the surface, the 100 meters. And we can see the pulse of alkalinity to the surface. And then we are learning because this is material that is in processes, because we are learning about how the water masses are transforming, who is pumping and what biochemistry to the surface, and which conditions we have in this nice process that occur in the Gulf of Mexico. And then just for finish, we have many lessons to learn, because we were talking about the coastal area when we have a local OA, but in the addiction, we have many things to learn about where the waters come from. Maybe we need more calculation about the Caribbean waters, the statistical seasonality, the incursion of the loop currents inside to the Gulf, how this volume of Caribbean water modulates the depth of the carbocline and upticline. I means all the biochemistry inside to the Gulf, depending on the volume of this incursion of the water. Another subject is about the soup water, how dissolved and the effects that we have, the homework that we have of the respiration effect in the wet side of the Mexican waters, and the effect of the OA. And this is just highlight that I really mentioned about the 20 years of data that we learned from the statistical, the Caribbean sulfur waters and the roles. And I just finished with this last message about the implication of that we learned about the Caribbean sulfur waters and the carbon system on the Gulf, in the Mexican area. I mean, also in the Gulf of Mexico or Gulf of America. Is what I have. I really want to say thank you for the invitation and to knowledge to the CONACYT, the center, for these two nice projects that we have with all these colleagues that we are working on it. Thank you so much.
Great, thank you very much for the. So I guess next is for Siobhan to have the audience ask questions.
Yeah, so if you're in the audience, there should be a panel where you can ask your questions. Feel free to type them in there. There's also an option to raise your hand. We've had some issues with unmuting, so you can try to raise your hand, but I think the best bet is typing the questions in the chat. We can give you guys a few seconds, but I can start off with a question that I had with the Fitobloom project, the most recent one. I was wondering if you guys during your sample time had sampled during any storms, or maybe what the weather was like during the transect within that core ring.
Yes, you know that the cruise was done in November, and this amazing cruise group, they have an experience that they never going to forget, because they have a north conditions, and then half of the cruise they have the north norte, we call it Mexico, right? The very strong winds, but this guy was working. There are a nice movie this guy developed just to see the effect and how the boat was shaking, and they lost one instrument, very expensive instrument, and well, was amazing condition, because as you say, this structure just alone have a biochemistry different masses, but in combination of this north half of the crust also spice the environment of the condition. And then the zoo have this sampling for this condition very special. As I mentioned, also we have a pulse of the river in one of the stations that we just catch up. The thermosal catch up this incursion of the low salinity water in one of the stations. But the discrete sample we were lucky to get, but it's more convenient when you see the thermosal. Okay, and then combination of the Mississippi River, the north, and just the biochemistry by own characteristics offer a cocktail of the conditions, yes, and a challenge.
Thanks so much.
So Martin, I do also have a couple questions. The first one is for this coastal acidification that you observed off the coast of Yucatan Peninsula, and also there's the influence of the loop current that can basically get rid of the acidification signal. So I'm wondering, I should have read the paper. So what is the frequency of the loop current influencing very close to the coast?
This is interesting because you know, I would like to tell you with all the elements, because I don't know if this is my answer. I'm not sure yet, but I imagine that based on the statistical observation that we have, when we have the maximum extension, that is mean summer, late summer, and early fall, and when we have the maximum extension. Something what we noted based on the ADT observation was the loop currents is more close to the peninsula, and when we have this minimum instruction, it's like decrease oxide. It's about right. Probably my answer, my guess is maximum because what I mentioned, the vection clean everything was in August. This sampling was in August when we have the maximum extension of the loop current. But it's just one sample, one cruise, and
sure.
it's a guess. It's a guess, yes. But probably the observation of the ADT, other satellite products, we can be more robust in these kind of questions based on the how close is the loop current to the peninsula of Yucatan, because we notice how it's moving this water. We put some particles simulation over the ADT, and we can see how we are moving over the shelf, just to have a better imagination of the processes, right?
Yeah.
Yes.
Great. So my other question is, you show that in the Western Gulf, there is a higher levels of DIC and lower level of oxygen or higher AOU. I'm just wondering whether this concentration difference between the Western Gulf and Atlantic is also because of the longer residence time when the water enters through the Yucatan Channel into the Gulf and then stay in the Western Gulf longer, so that's why there's accumulation of signals.
Yes, I agree with you. I guess it's something like that. My mind is there are two movies that can hypothesis, right? But can be mixed in both. It can be three. But one is, as you say, is the water going inside, and we have the resident time, and we have the process of the organic matter respiration that decrease the oxygen, increase the DAC, right? The other one is can be probably that the volume of the water is less the Caribbean sulfur waters in the western part, and we have more influence of the more deep water masses, like the TAC influence. Because it's interesting that this guy Gentil et al., they mentioned how this dissolution of the soup is dissolved and is mixing with the upper water, and they say the way that they suggest the Gulf common water is formed. But we don't have soup in the western side, and also the TAC is more close to the, is more shallow. And then I'm thinking that probably is some flavors from the TAC with the biochemistry stronger in the western side than in the east side. And then it can be, or can be the third one is, as you say, is the recent time plus these physical processes. We used to have in the Campeche the semi-permanent mesoceral circulation that is probably pumping more water from the deepest area, right? That can be this process.
Great. Yeah, thanks.
Yes.
That kind of leads into our question in the chat. They ask, is this study consistent with earlier work that the North Atlantic water preferably flows out of the Gulf in the loop current, and the Caribbean waters are preferentially caught in the loop current eddies? So it's more about the physical dynamics of the loop current.
Okay. Could you repeat? Because I confused about the Peruvian versus the Gulf.
Yes, sorry. So is this work consistent with earlier work that the North Atlantic water preferentially flows out of the Gulf in the loop current, and the Caribbean waters are caught within the loop current eddies?
Yes, I guess so. I agree with that. I guess this is something that is happening. Yes, of course we need the help from the physical oceanographers to be sure about that, right? And it's a challenge because you know, everybody is short distance between Yucatan and Cuba, and we have a Mexican part, and another portion is Cuba, and then it's a challenge for the physical oceanographer to put sensors and all these transit to have a better idea. And then you go to Florida, of course, it's from US, but it's a puzzle because as a US, of course, you have all the Florida channel, but we miss this other part. And then this is why I mentioned that it's a national challenge. We need the physical oceanographer from the 3 countries to do this job. Yes.
Awesome. Are there any other questions from the attendees? I think that might be it. Let's see. Yeah, there's no hands. So thanks for coming, all of the attendees that are still here, and a huge thanks to Martin for coming out and sharing his awesome work. And that's it from me. Shinping, do you have any other closing remarks?
No, well, yeah, thanks again, Martin. This is a wonderful presentation. I need to follow up on the papers. I read some of them, but then definitely not all. And so this also concludes our summer webinar series. And then, you know, look forward to seeing all of you, audience, in the fall, and we'll have more exciting talks coming up. Okay, thank you.
Well, thank you, both of you. I know that this new system, also we are learning how it's working, these virtual meetings.
Exactly.
We have the experience here in Ensenada that we have a meeting, and that people is over here, but they try to catch up this talk later, few days later, and then we need to say thank you for the people who's actually here with us, but also for the future people who will see this presentation. And then I want to say thanks to all of you from the present and the future.
Yep, thank you.